Hi, everybody. Good evening, good morning, good afternoon, wherever you are. Welcome to this first webinar of the International Academy for Clinical Hematology. I'm Mohamed Moti from the Sorbonne University in Paris, in France, and I'm the chairman of the International Academy for Clinical Hematology. First of all, happy New Year and all best wishes for a healthy and successful New Year. I hope you are keeping safe and doing well. I hope that we will be able to get rid as soon as possible of this pandemic, which is really which has generated a lot of losses. So we have some deep thoughts to all those colleagues, friends, unknown persons we lost over the last few months, all people suffering and struggling with this COVID-19. But thanks to our solidarity, thanks to science, thanks to the vaccines, we will make it all together. So despite the difficult environment here and elsewhere, we continue our educational activities under the umbrella of the IACH. The year 2021 will be another success. The activities of the Academy are really flourishing. And we didn't wait the COVID pandemic actually to develop all of these virtual and digital activities. I kindly remind you that our first webinar was in October 2018 when we have launched this series, and it has been amazing since. Every webinar is attended with several hundreds and thousands of people depending on the topic. But not only webinars, we have developed the activities. Remember, we have our peer-reviewed journal, Clinical Hematology International. It will be soon referenced on PubMed. We have celebrated almost 2 years, and it has been very successful thanks to all your submissions. This is a fully open access journal. It's not a predatory journal. You will not be asked to pay anything for submission, and there are no page charges. This is a pure academic activity. So please submit your best research papers, reviews, letters to the editor, to Clinical Hematology International. Of course, we have our monthly webinars, and one of them is the one you will listen to in a few minutes. But we have introduced a couple of months ago the superb activity entitled the ICH Journal Club. So we would select every 15 days actually a specific peer-reviewed article, and we will discuss this with a couple of experts. Usually one of them is a co-author, the other one is not a co-author, and this has been very, very successful, and thank you for being loyal to these activities. Of course, we do have the annual meeting of the IACH. This is now scheduled for early October 2021. We don't know yet if it's going to be a digital or virtual or an in-person, because it's very difficult to make predictions in these circumstances. But in May, 7th to the 9th of May, we have the 7th Congress, the famous COMI, Controversies, Worldwide Congress on Controversies in Multiple Myeloma. And this is really a highly successful activity, very well attended. So a huge number of activities. All of these activities are available free of charge, no registration to anybody. So this is really purely academia driven. And of course, we are able to achieve this thanks to the generous support of different pharma companies and donors. So thank you all for your support. And now, as it is a tradition every year, we have 2, 3, 4 special webinars dedicated to the ASH data. As you know, ASH is really the place to be. This is our most important congress in our field, and the amount of data being presented is huge. And this is why we aim every year between end of December, early January to provide you with some specific focused webinars where a top expert in this given field will do a sort of a comprehensive summary, will digest the data for you and try to capture the most important information, especially the information which will have some impact on a clinical practice. So I gave the summary about multiple myeloma, and this month we will have 2 big summaries, one dedicated to cellular therapy, actually delivered in 2 parts. Part 1 is today, part 2 will be roughly end of January, but also we will have in 2 weeks another summary dedicated to stem cell transplantation. So for today, we have the big pleasure and honor to have Professor Bipin Savani from Vanderbilt in Nashville in the USA. I don't need to spend a lot of time to introduce Professor Savani. He's really a top expert in this field, highly published, highly recognized, associate editor for BMT, associate editor for BBMT, which has changed name now, TCT. So this is a great initiative from our friends from the ASBMT and TCT, but he's also an editor in the British Journal of Hematology, and he has made really some seminal and very important contributions to the stem cell transplant and cellular therapy field. And he was the man, he was the guy who accepted really to go through all the cellular therapy abstract from ASH to put together for tonight a fantastic summary that I hope will be extremely helpful to all of you. So Bipin, thank you very much for taking the time, for making this huge effort, and we are very eager to listen to you. Of course, you will have the opportunity to ask questions using the chat box you have on the software, and Bipin will do his best to answer these questions, time allowing, of course, at the end of this webinar. But for now, welcome, Professor Savani, and wherever you are, again, Happy New Year, and please stay safe and keep well. Bipin, the floor is yours. Thank you very much.
Thank you, Professor Modi. Good morning, good afternoon, and good evening, my friends in Europe, Israel, India, wherever you're located. So I welcome you to part 1 ASH 2020 Cellular Therapy Summary. So today we're going to review ASH CAR T presentation part 1. Lots of good abstract. We'll try to provide you pertinent brief information on clinical abstract. I have no specific order on how I'm going to review all abstract today, but I'm sure you will enjoy. Abstract not selected for discussion today will be previewed in part 2 ASH summary in about 2 weeks. We can't discuss everything in 90 minutes. Special thanks to our friends, colleagues, well-wishers who provided their slide to discuss their data with you today. I have no conflict of interest to disclose. So if you recall, I reviewed a few months ago ASTCT, TCT 2020 summary for ISCH webinar series. And we did discuss this Lysosel data presented at the TCT 2020 meeting. Our friend Carlos Bashir presented in Orlando, our last in-person meeting in February 2020. Outpatient treatment with Lysosel in 3 ongoing clinical studies in relapsed refractory B-cell non-Hodgkin's lymphoma, including second-line transplant ineligible patient, Transcend NHL Auto1 study, outreach, and pilot. If you want to review this data, you can always visit ISCH webinar series for the details. Today, we are going to review ASH presentation, safety and preliminary efficacy in patients with relapsed refractory mantle cell lymphoma, receiving Lysosel in Transcend NHL Auto1 study. As you know, Lysosel is a BMS product, previously Celgene, originally Juno. Mantle cell lymphoma patient with disease progression after BTK inhibitor, very poor outcome. No need to go into detail. CAR T was recently approved by FDA for relapsed refractory mantle cell lymphoma. Approved product name is Tecartus, very well known to everybody here, a product of Kite Gilead. Lysosel, an investigational anti-9 CAR T product, demonstrated an objective response rate of 73% with CR 53% in about more than 250 patients from relapsed refractory large B-cell lymphoma cohort of phase 1 Transcend NHL 001 study. And we reviewed a few months ago, and it was presented in TCT meeting. So objective of current study presented at the ASH last month was to determine safety and initial anti-tumor activity of Lysosel in patients with refractory mantle cell lymphoma, and from the same study. Lysosel is administered as a standard separate CD8 and CD4 CAR T-cell component at equal target dose. Fascinating. CD4 and CD8 positive T-cells are selected from leukapheresis product by immunomagnetic selection, and they are activated, transduced, and expanded separately. The CD8 positive component is administered first, followed by CD4 component. The defined composition of lysosomes result in a consistently administered CD8 positive and CD4 positive CAR T-cell dose and low variability in CD8-CD4 ratio. Importantly, according to investigator when presented, defined dose and CD8-CD4 ratio of CAR T-cell may influence the incidence and severity of CRS and neurological event. Very interesting. Transcend NHL 001 is a phase 1 seamless design study, highlighted here, 2 disease cohort, large B-cell and mantle cell lymphoma cohort. ASH presentation was focused on the mantle cell lymphoma cohort. Bridging therapy was allowed. After lymphodepleting chemotherapy, Lysosel was administered at one of the 2 dosage levels. You can see here on your left. Administration and monitoring were allowed in either inpatient and outpatient setting, which is very important. Patient's eligibility criteria summarized here. Amended protocol, which is a later cohort, required patient to have a prior BTK inhibitors, alkylating agent, and anti-CD20 agent. Makes sense to include novel agent before you start CAR T therapy. Patient with prior transplant and secondary CNS lymphoma were eligible. And primary secondary endpoint standards phase 1 study design summarized here. At data cutoff, 44 patients undergone leukapheresis. 11 patients did not receive product, primarily because either they received Lysosel after data cutoff point or disease progression. 1 patient died. 1 patient did receive non-conforming product, meaning either CD8 or CD4 CAR T product did not meet the study requirement to consider Lysosel. And you can see many a time in your institution, non-conforming product. Total there were, and then we need to inform the FDA. Total there were 32 patients analyzed, majority in dose level 2. Slide summarize patients' characteristic, old cohort. Median age 67, 41 patients had blasted morphology. Median 3 prior line of therapy, prior BTK inhibitors in most patients. In short, very advanced mantle cell lymphoma, no good alternative option to control their disease. Main side effects were cytopenia, similar to observed in Lysosel study in relapsed refractory large B-cell lymphoma. 2 grade 5 events, means death, considered related to Lysosel treatment. One patient with high tumor burden had tumor lysis syndrome, and one patient had a cryptococcal meningoencephalitis. Both patients received dose level 2. No patient died from CRS and or neurological events. Grade 3 or higher CRS occurred in only one patient. Median time of onset of CRS was 6 days. Grade 3 or higher neurological events were reported in 4 patients, and median time to onset was 8 days. Duration was 4 days. For the management of CRS and neurological events, very few patients required intervention. Only 12.5% required both tocilizumab and steroid, 12.5% only steroid, and only 6% required tocilizumab alone. 34% patient, as you see here, had prolonged grade 3 or higher cytopenia, meaning cytopenia grade 3 or higher beyond day 28. Infection was present, but not much. Interestingly, severe hypogammaglobulinemia was present in 9%, which is not unexpected, but 7, meaning 22% patients received intravenous immunoglobulin throughout the study follow-up period of 2 years. Overall, 84% patient had objective response. 66 patients had a complete response. Median follow-up was about 6 months, and median time to first CR or PR was less than 3 months. Very effective. What about response in patients with high-risk feature? That's what we wanted to know. Summarize here at the bottom. Overall objective response rate, CR/PR, were similar in patients with high-risk features also. Median duration of response not reached after median follow-up of 4 months. To clarify more, response were generally similar in patients with bastard morphology and also in patients who were resistant to ibrutinib. Cellular side summarized nicely cellular kinetics. Median time of maximum lysosel expansion occurred in 10 days after infusion. Lysosel showed long-term persistent at 1 year in 67%. And in two-year follow-up, 33% of patients had a persistent lysosel. A small number, but very, very important information. I don't think I need to read this summary slide. Enrollment is ongoing at dose level 2, meaning 100 million CAR T positive cell in mantle cell lymphoma cohort. This is our next presentation. First in human study of allogenic anti-BCMA ALLO-715 CAR T-cell therapy and anti-CD52 monoclonal antibody ALLO-647 in relapsed refractory myeloma, universal study. Our center is participating in this study. Study sponsor is Allogene Therapeutics. Why this is moving faster? I have no conflict of interest. It's automatically moving faster. I don't know. Okay. I have no conflict of interest, but one of the authors, second last author, Arun, he used to be co-fellow with me. We used to co-fellow at NIH some time ago. This is a small size study, but very interesting concept and design. This is the first off-the-shelf allogenic CAR T-cell trial in relapsed refractory myeloma. Today, autologous BCMA CAR T therapy has demonstrated remarkable efficacy, but there are several logistic challenges limit to the access to auto BCMA cell therapy. One, allogenic construct has several advantages. Let me review here and summarize here, including minimal delays, Potential to treat all eligible patients within days without needing bridging therapy. Less product variability. Possibility of repeat dosing. And shown here, CAR T construct includes human-derived a single-chain variable fragment, SCFE, and 4-1BB co-stimulated domain. Generally, single-chain variable fragment-based CAR T-cell have an affinity for the target. Two key attributes, and I'll review soon in a minute. Two key attributes of this construct are talon-mediated knockout of CD52, which allows some selective lymphodepletion with ALLO-647, a CD52 monoclonal antibody, without affecting the CAR T cell, and talon-mediated knockout of Treg gene, which eliminated TCR alpha expression and minimize the risk of GVHD. Very fascinating. This is a phase 1 dose escalation multi-center study currently enrolling in multiple US centers. First patient was treated in August 2019, very young study. All patients received lymphodepleting chemotherapy with FCA, Abinese ALLO-647 monoclonal antibody. Multiple dosing regimens were used for ALLO-647, and ALLO-715, which is a CAR T-cell, was infused as a single infusion in an escalating dosing from 40 to 48 million CAR T positive cells, single infusion on day 0. This is a summary of patient enrolled when study was submitted for presentation. 4 of 35 enrolled patients were unable to proceed due to organ failure. 26 of total 31 patients included in this efficacy analysis. 5 patients treated but not reached the assessment point. Median follow-up is short, only 3 months, 3.2 months. Yes, yeah. Can see in here all heavily pretreated patients. I don't know, go into detail. Median 5 regimen. 94%, almost all patients were penta-exposed. That means patient has received at least 2 proteasome inhibitor, 2 IMiDs, and anti-CD39 antibody therapy. Most patients had prior auto transplantation. This is a safety profile. 40% patient had a CRS, no instances of neurotoxicity or no GVHD. 23% of patients had infusion reaction to ALLO-647 infusion, all grade 1 and 2. Efficacy data are reviewed here. You can see here with dose level 3, meaning 320 million CAR T positive cell, objective response was achieved in 6 of 10 patients with 4 VGPR or better in FCA higher cell dose group. Overall, 6 patients had VGPR or better response, and 5 of these were MRD negative. Very nice swimmer line plot here. Median time to response 16 days, increasing response rate as cell dose increases. 6 of 9 patients treated with dose level 3 or dose level 4 with ongoing response. Longest response, stringent CR at 6 months. Cell expansion was observed as early as 7 days. Very important. Improved expansion in patients who received higher dosing. Persistence of CAR T cell observed at month 4 in dose level 3. Author also mentioned that patients with CAR T expansion had a higher serum level of IL-15 at day 0 and day 14. Author presented one of the study patient case scenario, trial patient, to highlight the potential of this platform in patient with relapsed refractory myeloma. Patient has ongoing stringent CR at 6 months, and his patient is back to work. You can see on day 14 flow on your left side, more than 90% of peripheral lymphocytes were CAR T positive cells. Some additional details. on early expansion of CAR T positive cell, low level persistence over longer follow-up, and response over time. You can see nicely summarized graph. Again, I'm going to save time, not going to review this conclusion. Study is still ongoing with enrollment, and for planned evaluation of higher cell dose and lymphodepletion with FCA. This is our next presentation. Updated result from phase 1 CRB 402 study of anti-BCMA CAR T therapy, BB21217 in patient with relapsed refractory myeloma. BB21217 uses the same CAR molecule as BB2121, but is cultured with PI3K inhibitor BB007. That's why we got the extra 7. And goal is to enrich the T cell displaying a memory-like phenotype. CAR T cell, as we know, enriched with this phenotype may persist and function for longer than non-enriched CAR T cell. And as you know, persistence of functional CAR T cell after infusion may be one of the determinant of duration of response. Classical phase 1 study design, standard phase 1 primary secondary endpoint, eligibility criteria reviewed here. I'm not going to go into detail, all heavily pretreated patient. Standard study design, screening, leukapheresis, manufacturing. Manufacturing success was 100%, by the way. Lymphodepleting chemotherapy, BB21217 CAR infusion, day 15 bone marrow, and complete response assessment at 1 month. Total 74 patients collected, 2 withdrew, 3 patients infused CAR T after data cutoff point. So author presented nearly 69 patients data, median follow-up of 6 months. Decent number, decent follow-up. Patient characteristic and prior treatment history reviewed here. Median 5, median 6 prior regimen. 78% had prior auto. More than 90% patient had prior LAN, POM, Velcade, CD38. And 80% of patients were double refractory. 64% patients were triple refractory. Slide summarizes the safety and tolerability, mainly cytopenia, as we know, very common after CAR T. Grade 3 or 4 infection were reported in 18%. There was 1 death from infection within 6 months in the absence of myeloma progression. 2 deaths within 8 weeks of BB2127 infusion, both due to CRS. We'll review in a second in this slide. Slide summarizes CRS after BB21217 infusion. 70% patients develop CRS, but mostly grade 1, grade 2, as you can see in the table. Median time to onset was 2 days, some was early, and median duration was 4 days. Grade 3 CRS 1 patient, and grade 5, meaning death, 2 days, 1 on day 15 in the setting of grade 3 neurotoxicity, and 1 on day 6 with AFib and cardiac arrest. Very few patients required tocilizumab and steroid. Neurotoxicity was very limited, only 16%, mostly grade 1 or grade 2. 2 grade 3 events, 1 grade 4 treated with steroid and cyclophosphamide, and resolution, as you can see here. This is very important slide. Very decent overall response rate, 68%, CR rate, 29%. Median time to PR and high response was 1 month, and median time to CR or stringent CR was only 2 months. And it took as long as 24 months to achieve CR in some patients. Majority of patients who achieved PR or all CR patients tested MRD negative. Those who were tested were all MRD negative. After enrolling 8 patients in the expansion cohort as recommended phase 2 dose, to change the manufacturing process was implemented, slightly different. This change was implemented in order to align the manufacturing process as much as possible so that people don't criticize heterogeneity, especially FDA. So 29 additional patients were enrolled at 450 million recommended phase 2 dose and treated with drug product made using this updated manufacturing process, which is on your left. There was no obvious difference in patient characteristics between the group. In the group treated with original manufacturing process, median follow-up was 12 months, and 24 out of 40 patients responded, and 28 patients had CR or stringent CR. And the group on your left side, updated manufacturing process, median follow-up is still short. 19 patients were available for response. Out of 19, 16 responded, 84% response rate, and 32% CR or stringent CR. Median follow-up of 7 months, but impressive 17-month median duration of response. When we look at this response by the dose, median duration of response not reached in higher dosing infusion cohort, which is on your top line, green and blue line. Now, this is also important, persistence, expansion and persistence. Peak vector copy numbers, VCN, were correlated with response, shown here on your left. Persistence and detectable vector even after 12 months, 18 months, and long. You can see the red and green line here. Process of making BB21217 enriches the drug product for memory-like T-cell. Percent of central T-cell in BB21217 drug product is positively correlated with peak expansion, you can see here in the middle. Patients with sustained response are more likely to have drug product enriched for gene signature that indicates naive or memory-like T-cell, and gene signature that indicate presence of effector cells were depleted, which you can see on your left side. Fact that BB21217 enriches for memory-like phenotype suggests that extended duration of response is possible in the second cohort. Again, I'm not going to read this slide. Very interesting concept and design on the way. This phase 1 CRB 402 study has completed enrollment and follow-up is ongoing. I'm very impressed by the result. This is our next abstract. As you all know, FDA granted priority review to ID-Cell, Bristol Myers company, on track to deliver Cellular payday. ID-Cell is not approved for any indication in any geography at present. FDA approval of ID-Cell by March 3, 2021, possibly, is one of the required remaining milestones by the contingent value right issued upon close of Celgene acquisition in fourth quarter of 2019. And I hope it happens. And I expect this drug will be approved soon. I don't think I need to go into detail about relapsed refractory myeloma. ID-Cell demonstrated tolerability and promising efficacy in phase 1 CRB 401 study. Figure shows ID-Cell CAR design. I'm not going to go into detail, but as you know, targeting domain anti-BCMA, co-stimulatory domain 41BB, and T-cell activation domain CD3 zeta. Standard CAR T design. I'm not going to go into detail about process, evaluation. 62 patients treated on this study. Manufacturing success rate was 100%. Study now has median follow-up 18 months, with 18% patients still going on study, long follow-up. Patient demographic here. Heavily pretreated patient. All, as reviewed in prior slide, all patients had multiple regimen refractory to last regimen in dose expansion cohort. Bottom line, bad group of patient. Most common adverse event shown in the table on the right. As expected, cytopenia, very common in almost all patients. Grade 3-4 infection in 22% patient. Grade 3 or higher CRS in only 6%, and neurotoxicity in only one patient. 7 additional deaths within 6 months of study, majority due to progressive disease. Details of CRS after ID cell shown here. I think we reviewed already briefly. Higher risk with increased cell dose. Majority mild. Use of TOCI and steroid, very, very low. Details of neurotoxicity here. Majority, grade 1. Duration, 3 days only. Only one case of grade 4 neurological event. Less than 10% patient required intervention with steroid. Impressive response, very like, I love this slide. Overall response rate, 75%, CR nearly 40%. There was a dose-related response rate with higher dosing. All patients with CR or stringent CR were MRD negative by NGS. Slide reviews the duration response by dose and depth of response. Median duration of response, 10 months in all treated patients, and half of the 8 ongoing responders have duration response more than 2 years. Slide reviews duration response by subgroup. We want to know how they read with poor prognostic feature. Authors did not see any decreased response in patients with these poor markers, which you can see here in this slide. After median follow-up of 18 months, reasonably good follow-up for this kind of CAR T. Study showed median progression-free survival was 8.8 months and median overall survival of 34 months across all treated patients. Slide shows robust ID cell expansion with long-term persistence. ID cell peak within 1 month of infusion, remain persistent and detectable for many months post-infusion. You can see in this slide on your left. Patient with higher expansion had a deep response. You can see on your left. Updated results from CRB401 demonstrated deep and durable response with ID cell in heavily pretreated refractory myeloma. My personal guess, based on reading all newsletters, I think going to be approved soon. Our next presentation is Lumicar study, phase 1b2 study, fully human BCMA specific CAR T-cell, CT053 in patient with relapsed refractory myeloma. It's a product of CAR's gene therapeutic, a China-based company and headquarters in both in US and China. CT053 is a fully human BCMA specific CAR T-cell. CAR design reviewed here, nice figure. It is manufactured in 8 to 10 days, cryopreserved for administration, and product has already received FDA and EMA orphan drug designation. Again, I'm not going to go into study design, standard heavily pretreated patient, standard phase 1, phase 2 study design, standard procedure screening for SCS lymphodepletion, infusion, and everything. Patients and disease characteristics summarized here. Bottom line, all heavily pretreated patients. Half of the patients were penta-refractory. CT053 infusion and safety data reviewed here. Predominant toxicity, hematological as expected. 1 death, 1 of total 20. It was a sudden cardiac death, 4 months post-transplant, post-therapy, and considered not to be related to study drug. CRS occurred in 80%, but almost all grade 1 or grade 2. No patient developed grade 3 or 4 CRS. ICANS in 3 patients, only 1 grade 3. Very few patients required intervention. Very nice slide. Efficacy of CD53 demonstrated. Very impressive data. By week 4, nearly all patients had a significant decrease in their plasma cell. You can see here on the left. And importantly, a rapid bone marrow clearance of plasma cell observed in almost all patients treated at dose level 0 and dose level 1. Deep reduction in M protein during 1 month of infusion of CT053. Majority of patients had 25 to 50% decrease in M protein in first month. Figures show rapid depletion in both serum free light chain on your left, on your right, as well as soluble BCMA on your left. Very nice data. Slide showed data on CT053 expansion and persistence. That's very important to me. CAR cell became detectable in 3 to 7 days after infusion. And you can see a rapid expansion during first 2 weeks of infusion, with maximal expansion occurring on day 14. Author observed persistence of CAR T-cell for at least 6 months. It's on your left side. Duration of response. Among 18 patients with at least 8 weeks of efficacy follow-up after CT053, 94% objective response rate. 17 of 18 patients. 5 patients with CR and stringent CR. Response deepened after longer follow-up. Majority of patients had MRD negativity. The Lumicar study too, excellent data, relapsed refractory, small size study, but very impressive. No severe CRS, minimal neurotoxicity, only few patients require intervention, 94% response rate. Expect phase 2 study soon, phase 2 soon. Let's switch gear to leukemia a little bit. I'm not done with myeloma CAR study yet. Next abstract we are reviewing is presented by our friend in UK, Claire and Dr. Peggs, Professor Peggs. All CAR-19 updated data using Auto1, a novel fast off-rate CD19 CAR in adults with relapsed refractory B-ALL. As we know, prognosis of relapse ALL is poor. I'm not going to go into detail. Very few patients develop CR2, CR3, and despite that, allo transplant can't cure most of these patients. Novel agent, blinatumomab, inotuzumab, act as a bridge to the transplant. CD19 CAR delivered excellent response rate, but considered toxicity in elderly patients, but nothing approved yet for older patients, hopefully soon. Of the currently available CAR T, most are high affinity CD19 binder. This is very fascinating for me. But the Auto1 are low affinity binder with fast off rate. Author believes that conferring Auto1 with physiological T cell activation, it reduces the toxicity, it improves the engraftment, and potential long-term persistence to deliver long-term disease control, which is an issue in many of our patients. Slide highlights study design. Not going to go into detail. Lymphodepleting chemotherapy with slightly different cyclophosphamide dosing. Now, keeping elderly patient population in the mind, authors have developed some risk mitigation strategy. First thing, because elderly patients are high risk for developing all toxicity. First thing they do was they split CAR T into 2 infusions, day 0 and day 9, and then divided 2 groups based on tumor burden, blast percentage more than 20, or other group, lower burden. Patient with low burden started with high CAR T dose on day 0. Patient with high tumor burden started with lower CAR T dose on day 0. Day 9 CAR T dose listed here, but making sure there was no grade 3 or higher CRS or ICANS before second dose on day 9. Standard primary and secondary endpoints, all adult patients 16 to 65 years of age, relapse ALL, CD19 positive. No exclusion for prior recently approved therapy like blinatumomab or inotuzumab. 26 patients enrolled, 24 patients manufactured, and 20 patients treated. 4 patients died of progressive disease or infection during bridging therapy. 100% successful harvest resulting in a QP release product, meet all the requirement. Patient characteristics summarized here. All adult patients up to the age of 62, heavily pretreated. Median 3 prior line of therapy. 2/3 of patients had prior allotransplantation. Slide shows expansion and persistence of CAR T-cell. Very impressive. Initial expansion between day 0 and day 28, which is all merged and you can see up to 10 to the 6. Other impressive part of the study was ongoing persistent, even after 2 years after receiving product. You can see at M24 persistence. Safety profile, half the patient developed CRS, almost all grade 1 or 2. Very limited neurotoxicity. 2 died from progressive disease. 7 of 20 patients died on study. 2 from progressive disease, 1 from allotransplant complication, and remaining from infection. Half of them died within 1 month of infection. Efficacy, swimmer plot, very nice. You can see here after median follow-up almost 17 months, 11 of 20 patients are still in remission. Long follow-up, more than half, and these are all adult, elderly, ALL patients. Remember, you can't do anything with them, any treatment. Again, slide showed Kaplan-Meier graph. All patients and patients receiving a close manufacturing product, which is their later cohort. You can see here, excellent event-free survival, especially those latest cohort with close manufacturing product in red line. In summary, Auto1 product, tolerable safety profile. Response are durable and ongoing. CR observed beyond 24 months, supporting the development of Auto1 as a standalone therapy, and hopefully all these patients will never need transplantation. Next, we are going to review J4528, a Janssen product. Cartitude 1, phase 1b/2 open label multicenter study, evaluating efficacy in relapsed refractory myeloma. Very important study. Cilta-cel. I can't pronounce the long name here, so Cilta-cel is another name. Is a distinct second generation CAR T therapy, 2 BCMA binding domain. Author presented initial result, combined phase 1b and 2 Cartitude study of Cilta-cel. Study has a 2 portion, phase 1b to characterize the safety and confirm the dose, and phase 2 portion to evaluate the efficacy. Key eligibility criteria summarized here. I'm not going to go in detail. Here on left, you can search study design, standard CAR T clinical trial design. Target CAR T dose was 1 million CAR T positive viable T cell per kilogram. Here you see the consort diagram of 113 patients enrolled, 97 patients treated with Cilta-cel. Phase 1b portion had 29, phase 2 portion had 68, large size study. Median turnaround time for Cilta-cel manufacturing was about 1 month. No patient discontinued due to manufacturing failure. As you can see here, all patients were heavily pretreated. All patients were triple exposed. Almost all except 1 were refractory to their last line of therapy. As expected, all patients developed cytopenia, 10% grade 3 or 4 beyond 1 month, and 25% had grade 3 or 4 thrombocytopenia beyond 1 month, 4 meaning less than 10,000 platelet beyond 1 month. Grade 3 for infection in about 20% of patients. Slide summarizes non-hematologic adverse event. CRS, neurotoxicity, is our main area of interest. Let's look at CRS and there was no other major toxicity. Of 92 patients with CRS, majority, 95%. CRS onset, median time of onset was 7 days. Day 4 or later, 89% had CRS, those who developed. And day 6 or later, 74% of patients developed CRS. So almost 3/4 of the patients had CRS onset day 6 or later. This is opening the possibility of outpatient administration. So most patients, 99% patients had CRS resolved within 14 days. Slide reviews the neurotoxicity. 12 patients had other neurotoxin icons grade 3 or higher in only 2%. Other neurotoxicity grade 3 or higher in 9% of patients. Other neurotoxicities were resolved in 6 of 12 patients. One patient died from other neurotoxicity. One ongoing other neurotoxicity, the movement disorder, memory problem, those are the one I'm talking about, other neurotoxicity, but very limited, you can see here. Total 14 of 19 patients who received cilta-cel died. 5 due to progressive disease, 6 related to treatment related, which is reviewed here, which you can review later. Let's review the efficacy, most important. Response rate, almost all patients responded, 97%. CR plus VGPR, 93%. 67% stringent CR, quite exceptional. All heavily pretreated patients. Median time to MRD negativity, 1 month. Among patients with 6-month individual follow-up, most had a cilta CAR T positive cell below the level of quantification, 2 cells per microliter in peripheral blood, but response persistent. Progression-free survival, 12 months, 77%. Median progression-free survival not reached in either group, both overall and those who are CR-VGPR. Again, I'm not going to read this slide. Very impressive data. Outpatient administration is being studied in Cartitude 2 and Cartitude 4 studies. Let's switch gear again. Lymphoma, high-risk lymphoma. But this is a very interesting, fascinating study. Zuma 12, phase 2 study of axi-cell as a first-line therapy in patients with high-risk large cell lymphoma. Our program participated in this study. As we all know, current high-risk large B-cell lymphoma have poor outcome. I'm not going to details on this one. Authors presented updated efficacy, safety, PK result after planned interim analysis of Zuma 12. And I'm sure all you remember Zuma 1 data, which leads to approval of axi-cell Yescarta for the lymphoma patient. The long-term follow-up just recently published also. And 4-year overall survival is 44%. That must almost cure most of the half of the patient. Slide summarizes Zuma 12 phase 2 study design. To be eligible, patients has to have high-risk lymphoma, B-cell lymphoma, with MYC and BCL2 and or BCL6 translocation, or large B-cell lymphoma with IPS score C or more, anytime before treatment. In addition, have to have a 2 cycle of chemotherapy and positive interim PET scan. Study design reviewed here. I'm not going to go into detail. Phase 2 study primary endpoint was CR, and standard secondary endpoint listed here, which include CAR T-cells in the blood and cytokine levels in the CR. 37 patients enrolled, 32 patients received axi-cell, data cutoff August 2020. Median follow-up about little more than 9 months for both safety and efficacy evaluation. Slide summarizes patient characteristics. More than 40% patients were 65 or older, majority with advanced disease. Oldest patient you can see here, 86-year-old. I was on service. This is my patient, our patient at the Vanderbilt. I was on service when she received axi-cell. I took care of her from day +1 to day 14. After axi-cell, she is now 87-year-old and doing very well. Overall response rate, 85%, CR 74%, median follow-up 9.3 months, 70 with median follow-up of 9.3 months, 70% of patients with ongoing response as of data cutoff. Very impressive. Median duration response, median progression-free survival, median overall survival were not reached after median follow-up of 9.5 months. Very important, most common axi-cell grade 3 or high toxicity, higher adverse event were encephalopathy, 16%, ALT elevation, 9%, and severe neutropenia, 9%. 1 grade 5 adverse event occurred as a study due to COVID-19. CRS. All patients had resolved CRS. Let me give you the better information. Half of the patients received TOSI, 25% patients received steroid to treat CRS. Neurotoxicity, 69%, 25% had grade 3 or higher, which include grade 4 in 2 patients. Both grade 4 events were resolved, no grade 5 neurological event. Steroid required in 1/3 of the patient. Total numbers of CAR cell infused were comparable in Zuma 12 and Zuma 1 cohort. However, this is very interesting observation. Author observed higher frequency of central memory T-cell naive in Zuma 12 compared to Zuma 1, which is associated with a greater expansion of CAR T-cell as compared to Zuma 1, suggestive of improved T-cell fitness in first-line treatment. And you can see here level of memory T cells and naive T cell in pre-infusion product has been associated with favorable PK phenotype. CAR T expansion again was greater in Zuma 12 compared to Zuma 1, both peak and AUC first 28 days. PK profiles were both similar in both patients, double hit, triple hit, diffuse large B-cell lymphoma with IPI score 3 or more. So there was no different and PK profile between group, super high risk group or other group. Cytokines associated with CRS, neurologic events were listed here. You can see here both for neuro standard, nothing new, IL-6, alpha, gamma interferon, IL-8, CSF, everything. Very impressive data. My patient, 86-year-old, now 87 in remission, I can say that. Most important message for me was this study also provides new insight into pharmacology of axi-cell for patients exposed to fewer lines of therapy. I think that's a future. Let's review one more abstract and then we'll be done. We'll do 2-minute question answer. I have a lot, but I think can't do in all everything. We'll do in part 2 next. This is Zuma 5 study. Primary analysis Zuma 5, phase 2, axi-cell in patient with relapsed refractory indolent non-Hodgkin's lymphoma. Presented by Karen Jackson from Dana-Farber Cancer Institute. As we all know, patient with advanced stage indolent lymphoma, follicular marginal lymphoma, frequently relapse, standard treatment largely incurable, rarely leads to complete remission. And we know the Zuma 1 data, I think we all have memorized. And now we have a long-term follow-up, 40+% of patients overall survival at 4 years. Zuma 5 is for the relapsed refractory indolent lymphoma, follicular, and mantle lymphoma after 2 or more lines of therapy. Standard phase 2 study denied. I'm not going to go into detail, but large study, 151 patients enrolled. Relative large follicular lymphoma were relatively large compared to marginal lymphoma, and marginal zone cohort is still accruing. First 84 patient follicular lymphoma reached 12-month follow-up. Marginal zone lymphoma, 20 patients, at least 1 month follow-up. They were all included for efficacy analysis, and all patients were included for safety analysis. Key eligibility criteria reviewed here: 2 or more line of therapy, standard lymph node depleting chemotherapy, axi-cell dose 2 million CAR T positive cells per kilogram. Primary, secondary endpoint we already reviewed in previous studies. Total 150 patients, 51 patients enrolled, 146 patients received diffuse lymphodepleting chemotherapy. Efficacy analysis was performed in 104 patients, as I mentioned, at least 1 follow-up for marginal, 12 months for the follicular. Safety analysis in all patients, and let's review the data. You can see here all advanced stage disease, high tumor bulk, and heavily pretreated patients. 2/3 of the patients received 3 or more lines of prior therapy. In all patients with independent radiology review committee assessment, overall response rate was 92% and 76% CR. Among follicular lymphoma, 94% response rate, CR 80%. Among marginal rows, response was slightly lower, 85%, CR rate of 60%. Although it appears that response rate is lower among marginal zone lymphoma, this is largely due to the fact that 15% of patients were not found having measurable disease by central radiology review, even though the treating investigator thought they had the measurable disease. and were eligible for the study. Median time to first response 1 month, and look at the nice graph. No patient or disease factor were associated with response in this forest plot, nice forest plot. Notably, 9 patients found to have COVID. I'm sorry, not COVID, it's a COVID era, so I always think about COVID, but CD19 negative by immunochemistry. Let me repeat this sentence. 9 patients were found to be CD19 negative by immunohistochemistry. All 19 patients found to have response to CAR-T-cell. Very interesting. Duration of response shown here, median duration 17.5. For 64% of patients had ongoing response at data cutoff, and patients with follicular lymphoma, risks were ongoing in 78% patients who achieved in CR, 17% in PR. Patients who achieved CR more likely to maintain long-term response than PR patients. Follow-up of marginal lymphoma group is much shorter. Again, progression-free survival, 12-month progression-free survival was 74%, and 12-month overall survival was 93%. You can see here both graphs. Grade 3 or higher adverse events were common, 86%, majority of pancytopenia. Grade 5, 3 patients died, 1 related to CAR-T-cell, died from multi-organ failure in the context of CRS on day 7. CRS occurred in 82% of patients, very few, only 7% developed severe CRS. Only half of the patients required TOCI, and only 50% patients, 17% required steroid to manage CRS. Very good. Neurological events, any grade in 60%, grade 3, 4 in 19%. Surprising to me, incidence was higher in marginal zone lymphoma compared to follicular. 41% patient in marginal zone had a grade 3, 4 neurological event compared to 15% with follicular. 31% patient received corticosteroid in follicular lymphoma group to treat the neurotoxicity, while 2/3 of marginal zone lymphoma patients required corticosteroid to neurotoxicity. Median duration was 7 days. Median time to onset was 7 days, and duration was 14 days. Median time to peak anti-19 CAR T-cell level after CAR-T-cell was 9 days. CAR T-cell expansion by peak and AUC trended higher in patients with marginal lymphoma, could explain higher CRS or neurological events in marginal zone lymphoma patient. Most patients with evaluable sample, 78%, had low level of detectable CAR gene marked cell at 12 months. That's very interesting. Night slide shows association between CAR T-cell expansion and clinical outcome. Peak CAR T-cell level was associated with both grade 3 and higher CRS and neurological event in both follicular and marginal lymphoma, although marginal lymphoma has a short follow-up and a small size. Median time of most serum analytes was less than or equal to 8 days, mainly resolving by week 4. You can see here a level by day 0, day 3, day 7, day 2, week 2, and week 8. Medial fold increase in K analytes privilege shown by associated with, the rapid rise is associated with toxicity following CAR-T-cell infusion appeared greater in patients with marginal zone lymphoma, and again explains the more neurotoxicity and CRS in marginal zone lymphoma patients. In patients with follicular lymphoma, peak level of key serum analytes were associated with grade 3 or higher neurological event, and similar observation in patients with marginal zone lymphoma. In summary, CAR-T-cell demonstrated high rate of durable response with overall response rate of 92% in patients with indolent non-Hodgkin's lymphoma. Safety profile was manageable and appeared favorable in patients with follicular lymphoma compared to previously reported large B-cell lymphoma. So I'm keep going now, one more abstract, just to make sure we have enough covered. And this is all recorded session, so you can review it anytime. So this is my last abstract presentation for part 1 today. CD22 directed CAR T therapy mediate durable complete response in adults with relapsed refractory large B-cell lymphoma after failure of CD19 directed CAR T. That's very important. There are patients who fails CAR T-cell, CD19 CAR T. What do we do with them? And also, study addressed the response rate in patients with adult patients with relapsed refractory B-cell ALL. Abstract covers both lymphoma and ALL. My favorite group presenter, John, first author, Laurie, last author, Dr. Mikhalos, and my former NIH attending, Crystal McCall. So CD19 therapies have achieved durable complete remission in 40% of relapsed refractory large B-cell lymphoma. I think we have already memorized recent HC-cell long-term follow-up publication. But CAR-19 refractory large B-cell lymphoma has extremely poor prognosis, no doubt. We can treat again with CAR-19 theoretically, second round of CAR-19, but 30% of post-CAR-19 relapses had CD19 loss or decreased CD19 expression, big problem. And this presentation tried to address this major issue in our clinical practice. And this is regarding relapsed refractory ALL. CAR-19 therapies have achieved high initial CR rate up to 100% in pediatric and young adult relapsed refractory ALL. The relapse remain a significant problem, limiting durability of remission following CAR T therapy. I refer to Dr. Crystal McCall's presentation. I think I have one of her slide. Let me see. Yeah, this is the one. Look at this slide. So these are all CAR T patients, and those patients who did not receive CAR T followed by allo, look at the outcome. All patients, almost all relapse, no allo transplantation. So this is the big problem. Again, like we have seen in relapse after CAR-19 in large B-cell lymphoma, 20 to 95%, remember, up to 95% of post-CAR-19 relapses had CD19 loss or decreased expression, suggesting that antigen escape represents the major, if not the predominant mechanism of relapse in ALL. Also similar to lymphoma patient. We just reviewed this slide. So what do we do? Given that poor prognosis of patients relapsing after CAR-19 therapy or transplantation, there is an urgent unmet need for effective therapies in this population. So let me remind you, I think this is our 90-minute presentation. So we will be ending at 2 p.m., 12:30 to 2 p.m. So I have a few more abstracts to review. Thank you for reminder. Back here, CD22 is highly expressed on surface of B-cell, including on B-cell lymphoma and B-cell ALL. This makes an excellent CD22 as a target for CAR cell therapy, CAR-22 therapy. In phase 1 dose escalation study of 55 pediatric patients, CAR-22 produced CR rate of 70%, 61% MRD negative, this is a pediatric patient. More than 2/3 of patients in this group had prior allo, and almost 90% of patients had a CAR-19. I'm sure we all seen this paper, published recently in JCO by Nirali Shah, one of my former NIH co-fellow. It was published in JCO, as you know, 2020. So this encouraging early result in pediatric patients provided the basis of this study expansion in adult population with relapsed refractory B-cell ALL. Phase 1 study enrolled in parallel for both CD19 CAR-19 refractory large B-cell lymphoma, and B-cell ALL. Both were enrolled in parallel. Patient underwent all the standard leukapheresis after enrollment, but the manufacturing here utilizing Miltenyi Clinimax Prodigy platform, which was common at NIH. I remember when I was there too. Cell harvest was performed on day 7. For lymphoma cohort, 3x3 dose escalation design in order to determine optimal CAR-22 dose. For ALL, there was a fixed dose based on prior pediatric study, which was published recently. Standard lymphodepleting chemotherapy. Author presented data on dose level 1, that is 1 million CAR positive cells per kilogram. Initial assessment on day 28 and subsequent on day 90 and every 3 months thereafter. Primary endpoints were toxicity, manufacturing feasibility, and recommended phase 2 dose and safety. Key secondary endpoints are listed here, including clinical efficacy, CAR expansion, and persistence, and others. Key eligibility criteria for both large B-cell cohort and B-ALL cohort reviewed here, accruing parallel. For large B-ALL lymphoma, for large B-cell lymphoma includes prior CAR therapy, one of the eligibility criteria, or CD19 negative disease. For ALL patient, relapsed refractory disease could have isolated CNS or extramedullary involvement included in this study, which is excluded in many studies, but they included here. Patient must be more than 3 months since last dose of inotuzumab for obvious reason, as we know. And both group required CD22 expression at any level by flow cytometry or immunohistochemistry, and it is required prior to enrollment. All those ALL patients who received CD22 directed therapy, all relapsed refractory nowadays received inotuzumab. It required to check CD22 expression post-treatment. And in any patient who received prior CAR therapy, to assess circulating CAR T-cell, CAR cell in the peripheral blood by flow cytometry to make sure numbers are not too high. It should be less than 5% in order to be eligible. You don't want 2 CAR T-cell to fight there. Small size study, but very important, very interesting study. I would say top class study. Total 9 patients received CAR-22, 6 B-cell ALL, 3 large B-cell lymphoma. Like we discussed before, all dose level 1 data reviewed here. CAR-22 was manufactured in all patients. Median time from leukapheresis to infusion was 15 days. Slide reviews the patients and disease characteristics. Median age in B-ALL was 43 years, up to 62 years of age. Median age in large B-cell lymphoma was 53 years, up to 57 age in this cohort. Among ALL, all with high-risk disease feature. All patients had prior allo. Remember, 2 out of 6 patients had prior CAR-19, and 3 had received CD20T directed therapy. I'm sure it is inotuzumab. And 5 of 6 patients had prior CD19 directed therapy. Assume it was blinatumomab. In lymphoma, every patient had at least one high-risk feature, and all patients had prior CAR-19 therapy. No grade 3 or higher CRS, no neurological events. Small study, but gives important information. 2 lymphoma patients and 3 ALL patients developed grade 2 CRS. Resolution of CRS occurred within 24 hours of intervention. Looked like all 5 received toci and steroid. Persistent cytopenia was a problem. Occurred grade 3 or higher in 5 out of 9 patients, not uncommon. Slide reviews large B-cell lymphoma clinical outcome. All patients achieved response at day 28. Median follow-up of 10 months, and all patients are alive and in CR. All patients had prior CAR-19, as you remember, the eligibility criteria. With median follow-up of 10 months, all patients are alive and in CR. Very impressive. Not that good for ALL, but there are some reasons for that. In ALL patient, achieved response by day 28, all patients. 5 patients achieved MRD negative, 5 out of 7 MRD negativity by flow with limit of detection 10 to the minus 4. 3 out of 5 patients were MRD negative by NGS with limit of detection, as you know, 10 to the minus 6. However, after median follow-up of 3 months, 5 of 6 patients had disease relapse. At the time of last follow-up, 1 patient who achieved CRI at day 21, day 28, he has remained NGS negative remission at 4 months. Now look at this, is interesting why I was assumed they were all relapsing, some relapsing. All patients who relapsed had the repeat disease assessment to evaluate CD22 expression. 3 patients, 3 out of 5 who relapsed, meaning had demonstrated absent or significant decreased CD22 expression by flow when compared to enrollment. That could be one of the explained reasons. CD22 expansion and persistence were repeatedly measured using multi-parametric flow cytometry assay. Both group lymphoma and ALL group showed robust expansion, you can see on your left. Expansion of CAR T, CAR22 with peak level at day 14 after infusion. Predominantly, it's very important, CD8 positive CAR22. CAR22 positive cells are detectable up to 6 months post-infusion. You can see here in both groups. Author also presented interesting CAR22 versus CAR19 expression and persistence for both CD8 positive and CD4 positive CAR19, CAR22, their own data. Peak level of CAR22 was 12 and 56-fold greater than CAR19 level, shown in the red here. Very different, widely apart graph. In short, early efficacy, reliable rapid manufacturing, and manageable toxicity identifies promising role of CAR22 therapy in treating patients with relapsed refractory ALL or CAR19 refractory large B-cell lymphoma. I think we have time to review one more abstract. This is the title, presented by my friend Nitin Jain from MD Anderson. Preliminary result of BALL-01 phase 1 study, UCART22, allogenic engineered T-cell expressing anti-CD22 CAR in patients with relapsed refractory CD22 positive B-cell ALL. This is a Cellectis product. Stock is going well. If you remember, look at the symbol, Cellectis. Stock has doubled in past 6 months. UCART are off-the-shelf allogenic product whose production can be industrialized, therapy standardized with consistent pharmaceutical release criteria over the time and from batch to batch. So consistency of the cell, just take it and give, and repeatedly we can give as if needed. As we know, prognosis of patient with refractory ALL is poor. I'm not going to go into detail, but we have a limited option. I need something good in near future. So authors have previously published UCART19 data in ASH 2018, I think, using lymphodepletion regimen combining cyclophosphamide, fludarabine, with or without alutuzumab, FCA lymphodepleting chemotherapy. Full data of 20 patients showed in that study acceptable and manageable safety profile of UCART19. Severe CRS in only 15%, only 2 grade 1 cutaneous acute GVHD, no severe neurotoxicity, and in this heavily pretreated population, 88% patient, evaluable patient, 14 of 16 patients achieved a CR or CRI, most MRD negative. Very interesting data, 88% good response. All patients who achieved MRD negative had evidence of UCART19 expansion. So with this background, authors presented UCART22 data. UCART22, as I said, allogenic off-the-shelf CAR T product. It's immediately available, standardized, manufactured at large scale. Ability to redose patient as needed. Here, CAR T expression redirects T cell to tumor antigen CD22 instead of CD19, you CART19. T cell receptors are knocked out using TALEN technology to eliminate the risk of GVHD. We already discussed TALEN technology when I presented earlier allogen therapeutic product, AlloCAR T in myeloma. CD22 is disrupted using TALEN technology to eliminate the sensitivity to alemtuzumab for infused CAR T-cell. So no downside, CAR T-cell preserved, but no GVHD. Slide shows in vivo anti-tumor activity of UCART22. UCART22 extended survival of immunocompromised mice engrafted with Doddy cell line, which is CD22 positive Burkitt symphola cell line, in a dose-dependent manner. Nice publication. This is a study design, key inclusion criteria, age up to 70, so we're good now. Patient with relapsed refractory CD22 positive B-cell ALL, plus CD222 expression required more than or equal to 90%, received at least one standard and one salvage regimen, very reasonable eligibility criteria. Endpoint include safety, tolerability, assessment of maximum tolerated dose, and recommended phase 2 dose. UCART22 expansion and persistent and immune reconstitutional study. Trial started with dose level 1 after FC lymphodepleting chemotherapy. Currently, trial has been amended to add alemtuzumab to FC at a dose level 2, and this cohort is currently accruing patient. Nicely reviewed the study events. Everything is perfect, excellent, like we see in almost all patients, screening, enrollment, infusion, dose limiting toxicity, observation, disease evaluation, and long-term follow-up. Small size study, but impressive concept and design. I have already highlighted impressive company's UCAR19 data before, ASH 2018 presentation. 5 patients treated with UCART22 so far, 2 in dose level 2. Baseline characteristic reviewed here. Heavily pretreated. Most patients received multiple therapy, including 2 patients had prior CAR19, relapsed refractory at study entry. No patient experienced dose-limiting toxicity, CRS in 3 patients, 2 were grade 1, no patient required tosu or steroid. No patient discontinued treatment due to UCART22 related treatment emergent adverse event. And this is very good. 2 patients at day level 1 achieved CRI at day 28, and one of them attained CR MRD positive at day 42, and subsequently received transplantation after receiving inotuzumab. One patient at dose level 2 had a significant bone marrow blast reduction at 28, and then patient progressed. Host T cell reconstitution observed in all patients within dose-limiting toxicity observation period. You can see here nicely on your right. We expect more promising data in future. FCL lymphodepleting cohorts are enrolling patients. Very fascinating, and I'm very impressed by the initial result and the preclinical data and UCART19 result in the previously. So let's stop here for this part 1. Friends, thank you so much. Special thanks to our friend, colleague, and well-wishers who provided their slide to discuss the data with you all today. Summary webinars will be available on ISCH website. Also, direct link to YouTube videos. You just Google ISCH webinar series and you will get it. Goal is to share the information to help our patient, and I thank you all for your patience. Now I'll be happy to answer if you have any question, and we will meet again for part 2 to discuss remaining CAR T abstract presented at the ASH meeting. Again, thank you so much. Please send your question via text messages in the chat section. And if I don't know the answer, I know how to contact the original presenter speaker. Arnold, it looks like we are done.
There are some questions.
Okay. Where can I find the question? Yeah, okay.
So maybe Professor Savani, I'm still following. I can help you with the questions. So one question: how to choose between BiTEs and CAR T-cells against CD19 in ALL and other lymphoid malignancies, for instance?
Very, very difficult question. And I don't know the exact answer. But the one thing I can say that BiTEs are very promising and not gone. So what we are going to do, one of our fellow here, Amit Patel, Dr. Dolaria, we are reviewing all ASH presentation on CAR T and BiTE. And we're going to compare and see what we can say. But I don't think I can answer this question. If you have a comment, please, Dr. Professor Modi.
No. I'm just a moderator here. So
I don't.
we have a question about Zuma 12, where the results are really impressive. So would you be willing to consider to move forward these CAR T cells into the frontline setting?
I think so. And after looking at my patient, she was 86-year-old, and there are a few others. But yeah, very impressive data, and not much toxicity. I don't know how many patients require ICU transport. I don't have those details. We need to look at the morbidity also. And I think maybe to consider cost factor analysis also when we are using this upfront most expensive therapy. But yeah, very impressive data. And to me, that's a future, I guess. Once we know that CAR T cells are not that much expensive, and once we have many computers are coming out with less toxic CAR T cell therapy. You can see, I was so impressed by many CAR T presentation and the way they modify the amount of central memory CAR T positive cells and CAR T naive cells and decrease the toxicity, keeping persistent and good expansion for a long time. So that's a future. I would say, yeah, it is coming for upfront CAR T cell therapy for many hematological malignancy. Now, I do have many myeloid malignancy and CAR T, which I'll be discussing in part 2, but all on the way, very impressed. But at the same time, I would say BiTE are not gone yet.
We have actually plenty of questions about CAR T cells in myeloma, but I see time is running and we'll have to close. So one last one. Would you be using, if possible, frontline CAR T cells in high-risk multiple myeloma, which I think is a really very good question.
Yeah, that's my. It's a political answer. People will kill me if I do something wrong because there are lots of companies are listening to this conversation. But my soft answer is yes.
Oh, amazing! Thank you very much, Professor Savani, for this.
I didn't want to create big controversy by answering in detail because it's still very sensitive issue right now. Which one we go upfront?
You are absolutely amazing. Thank you very much. It's an impressive summary, 90 minutes of the best of ASH in terms of CAR T cells. And part 2 is yet to come. So yeah, thank you all for joining this webinar of the IACH. Wherever you are, I wish you a good day, a good evening, a good afternoon, and please stay safe and keep well. Take care. Goodbye.
Thank you. Thank you. So in part 2, we are going to discuss transplant after CAR T relapses, myeloid malignancy, and so and so.
Excellent. Take care, guys.
Thank you, sir.